So, hello everyone. I'm really excited to have you all here tonight and especially to be welcoming Diana. Diana, where are you joining us from tonight? Are you in the Galapagos, Ecuador?
Yes, I'm in San Cristobal Island in the Galapagos.
Wonderful. I met Diana, it's been 3 years ago today. Well, not today, 3 years ago now, at Sharks International. And she does some amazing work in the Galapagos and on genetics in sharks. And I'm going to let her introduce herself a little bit further and take the show away, really. So we've got a really exciting talk. She's going to give us a really exciting talk. And I will be fielding your questions through the Q&A. So if you have any questions, pop them in the Q&A and I'll be asking Diana at the end. Diana, thank you so much for joining us and I'll let you take it away.
Thank you, and thanks for inviting me. I'm so excited to see you again. Let me just share my screen for a little bit. Oh, I cannot. Could you let me share screen, please?
That should have worked now.
Perfect. Let me see. Does that work? Can you see it?
Yes, perfect.
Great, right. Well, thank you so much for the invitation. I am super excited to be here and to share with you a little bit of what we do here in the Galapagos and to talk to you about one of my favorite topics, which is using genetic tools for conservation and more specifically now for shark and ray conservation. But before we go any further, I want to tell you where I am exactly at the moment. Jenny just mentioned it, I'm in the Galapagos now. Galapagos are this group of islands that are about 1,000 kilometers from the Ecuadorian mainland, and this is in South America. And I am particularly in this tiny island here, just below that fish, that it's called San Cristobal. And these islands are special in many ways. They are very biodiverse. They hold a large amount of endemic species, so species that cannot be found anywhere else in the world. And we are part of this region that's called the Eastern Tropical Pacific. So somehow we're connected with other countries and with a lot of species, especially the ones that migrate a lot, the whales, turtles, sharks, manta rays. So when we study the Galapagos, we have to keep that in mind, that we're part of this big system. And I have put up just a few pictures here for you to see what a daily field trip looks like when we're out studying sharks and rays. So this talk is about genetics, but I don't want to make it any complicated, or I want to keep it very, very simple. But we need to understand just a very few concepts. And one of these, of course, is genetics. What is genetics? Well, imagine you're going to build a house. Then the first thing you need is a blueprint with precise instructions of what to do and how to do it. And then you also need a lot of materials like bricks or blocks that will help you get to that house, right? So something very similar happens with living organisms. All living organisms do need these precise instructions as well. And this is what the DNA is. The DNA holds all this information or all these instructions in order to build organisms. And the building bricks in this case are something we call nucleotides that are represented by letters and that all together form this genetic code, right? So no matter if it's a shark, if it's a ray, if it's a turtle, if it's a lizard, if it's a human, we all have DNA. But one of the first things that we want to know when we study the genetics of wild animals is how genetically diverse they are. And what does this mean, in other words? In other words, being genetically diverse means I am capable to adapt. I am capable to survive, and I have enough tools to do so. So is it important? Of course it is. Imagine you're on a trip with your family, with your friends, and you're driving your car in a highway, and then your tire goes flat, right? You pull on a side and you go to the back of the car to see if you have tools. You find a toolbox. What will be more useful for you? To have a toolbox full of different tools, different type of tools that will help you solve the problem, or have a toolbox that it's full of the same tool. So a lot of tools, but all the same. What will be more useful? The first one, right? Something very similar happens with the genetic code. The more genetic diversity we have, the more tools we have to solve problems or to face changes in the environment. So the more chances we have to survive, right? So I'm going to, with these little concepts, I hope everything it's clear so far. I'm going to tell you how a daily field trip looks like for us and how is it that we get to this part of the genetic studies. So can we get DNA from sharks and rays? I already told you every living organism has DNA, but how we do that? Well, first thing, we have to wake up very, very early in the morning and go to the field. These are a couple of colleagues of me. We deploy a net. This is a day, and this is particularly for a project where we monitor baby sharks, baby blacktip sharks. So we deploy this net. We always have a person patrolling the net that's on the water. You have here the guy with the snorkeling set. So we leave the net for a little bit, and when a shark comes and meets the net, this person who's patrolling all the time will take it out of the net and bring it to us, right? Has to do it very carefully. We're handling a wild animal. We don't want to harm the animal, and we don't want the animal to harm us either. So it does it something like this, keep it making an A-shaped movement so the water keeps flowing through the gills of the shark and it can still breathe normally. This person who's in the water then will pass the shark to someone that's on the boat or on the beach, depending on the place where we are doing the monitoring. So the person who's in the boat is ready, and when we take this baby shark out of the water, we make sure we check the time. We don't want out of the water. We don't want to stress. We want to keep it safe over anything else. So once we take it on board, we have there prepared some conditions. We have this little roof to make sure there's not much sun coming through and the animal's not getting too warm. We have this little pool-like structure where we put a little bit of water as well to keep it cool. And then we start taking and recording a lot of measurements. We see the total length of the shark, the length in between fins. We do the girth of the shark. We record the weight. So we have a scale on the side where we put it. We check if it's a male or if it's a female. We see how young it is. When they're very young, when they're newborns, they have the umbilical scarf still a little bit open. So we record all this data. And then one of the last things we do before we release it is taking a tiny little bit of fin clip. How we do that? How big is this fin clip? Imagine the nail on your little finger. And now look at your nail. Half of your nail, that'll be the size of the fin clip we take. And we take it from the dorsal fin of the shark. So it's a tiny little bit. It doesn't hurt the animal. And then we put it into this little tube that is full with alcohol. We do this because the alcohol preserves very well the DNA. So when we take it to the lab later on the day, we will be ready to process it in the lab. So the last thing we do in the field is releasing the animal. We also are careful on taking the time, how long was the animal on the boat, and then we follow them up. So we have someone else on the water just swimming around, making sure for at least 5 to 10 minutes that the animal it's okay, that they are not having any problem. And then that's, as I said, how the day in the trip looks like, right? But then what? Then we go and start with the genetic part. So we take these samples to the lab. In the lab, we extract the DNA. Remember, I told you what the DNA is. We extract the DNA. We make sure at every single step that we are doing things right. We have to be very focused on what we do. We amplify some specific regions or areas in the DNA that are of our interest. And then at the end, we sequence this DNA. What does that mean? We come up with the strand, with these letters I told you, right? The nucleotides. So we end up with long strands of DNA and all these letters. Then we are able to compare one to another to respond a lot of questions. And now I'm going to tell you some of the questions we are responding with genetics. If you have any question, you can just tell me. But one of the first things we're doing here, let's imagine we are a group of 10 or 20. And in this group of 10 or 20, we have some siblings. We have 2 or 3 pairs of siblings, right? With a genetic code, I can identify who are the siblings, who is more related to one another versus the other people in the room. So we're doing something similar with these sharks. So these images, these pictures that I show you were of the Galapagos, Blacktip Shark, right? This is a photo of the Blacktip Shark. So this is the San Cristobal Island that I told you I'm based on. And there are many places around San Cristobal where we are seeing these baby sharks. Now, the big question for us is, are they all connected? Are they all related to one another? Are they all family? Or we have different families around the island, or we have different groups that do not meet and do not encounter each other. So of course we cannot follow them up 24/7, and we cannot just go and swim behind a shark for 2 months in a row. But what we can do is take this tiny piece of tissue and then do the DNA extraction and test with the DNA if we are seeing multiple families or not. So these stars that you see here around these islands are the points where we are taking the sharks from. And we're checking how many siblings we find in between one place and the next one, and seeing if there's any difference on being on this side of the island versus being on the other side of the island or not. Now, it's difficult to imagine the scale of this island here, but just so you have an idea, if I take a boat from here and try to reach here, it will take me about 2 to 3 hours, depending on the boat. So there are places that are a little bit distant. And this study that we're doing with the blacktips, we are combining with other techniques as well. So often when we apply genetics for study something, we also do other techniques that are non-genetic. In this case, we are using drones as well to monitor these bays, these areas. So this is a photo taken by one of our students, Lauren, and this is in the same place where we are doing the sampling that I show you in the pictures. So what's the idea with these drones? That not only we're checking them one by one when we take them out of the water, but we're trying to make an assessment on how many baby sharks are there at a single time in one particular location, just to see if they're being affected by some factors. In the islands, we have local fisheries, we have a lot of tourism, and that can have an impact and that can have an effect on these areas. Other question that we're trying to respond, and this is not for sharks, but for the giant manta ray, is how are they connected in the Galapagos versus other populations in the mainland in South America. In Ecuador, we have an island that's called Isla de la Plata, which has the biggest, the largest aggregation of mantas in the whole world. So there are a lot of manta rays here, but we're also seeing the manta rays in the Galapagos. So the big question is, are they the same? Are they coming from Isla de la Plata to Galapagos and then going back? Or we are seeing two different families, two different populations there. And so far, we have found something very interesting, and is that the ones that we see in the Galapagos are not moving to mainland Ecuador. So they are staying here. They like to stay here. And that is something very interesting. It happens with a lot of species. It happens with orcas, it happens with sea lions. They do like to stay in the Galapagos because they have plenty of food here. They have a space that is safe here. So they like to stay. They don't go too far away from the Galapagos. And for the manta rays, the sampling, of course, is a little bit different from the little sharks. We cannot just hold a giant manta ray and put it on the boat. What we do in this case is we take this tiny piece of tissue, of skin, using a pole. So this pole has what we call a biopsy tip. So a little tip that's a bit sharp. So we just poke it gently and we get out the tiny bit of tissue. They are so big that they don't even notice when we're doing this. But this, I know it's not the best picture, but this is how the work looks like with the manta rays. And other thing we're doing with rays is with the spotted eagle ray. You probably have seen this before. This ray, it's distributed. So we find it in Costa Rica, we find it in Ecuador, we find it in Colombia, we find it in different parts of the world. But what we're not sure is what species exactly it is, the one we have here in the Galapagos. So it's been assumed, everybody thinks, yeah, it has to be the same that you find in Ecuador. It has to be the same that you find because it's close enough. But the thing is that we're finding that the ones that we see here in the Galapagos are not the same that we see in other parts in Central or South America. They are a different type of animal. So that's very interesting because if we want to preserve, if we want to conserve and to protect nature and to protect the animals, we need to understand, first of all, what is it that they are. So if we want to protect them, we need to know exactly what species it is because each species has its own particular characteristics. Ones will grow bigger, ones will occupy mangroves, ones will occupy rocky areas. So we need to understand that first. And then we can come up with a plan that helps us protect them. And one other thing that we are doing with these rays here in Galapagos is studying the golden, not eagle ray, the golden ray, sorry. And I have a short video here for you. Let me just hit play. This is also a group of baby golden rays in this area here in southern Isabela. The largest island, it's called Isabela. And this is a group of baby golden rays. So before we started with these projects, we didn't know that the golden rays were having their babies in some of the bays here. So that's something new we have encountered. And now this, why this is important? Because the nursery areas, the areas where the babies are crucial for the survival of the species. Sharks and rakes take a very long time to be mature and to be able to reproduce and have offspring. So when they do, the offspring, the babies, are crucial to maintain, to protect, so they can grow old and reproduce again. So these, what we call these nursery areas, are very, very important. And now that we know that we have nursery areas for golden rays, for eagle rays, for blacktip sharks, for hammerhead sharks even, these areas are a priority, become a priority for conservation for the local authorities. And this is what we're helping to do. By providing this information, we're telling them, look, here is where you have to give priority to protect, and this is why, right? And we are also combining this with other type of mechanisms. I told you, with other type of methodologies. and I'm going to tell you about this combination. We are using what we call BRAVs, which is a short for baited remote underwater videos. So what is that? We have this structure, we put a camera, and we put something with bait on a long arm. So we deploy this underwater, and we are basically calling everything that's nearby and that gets attracted by the smell of this bait to come, and it's automatically recorded in the camera. So this is a really cool technique, because in this case, it helped us detect a nursery for hammerhead sharks in an area that where we didn't know it existed. We knew there were adult hammerheads nearby, but we didn't know this was a nursery for hammerheads. And every time since, we have been coming back month after month to deploy the cameras, and we are constantly seeing baby hammerheads. And the hammerheads are a species that's endangered, so it's important to identify these areas where they're having the babies. And we have also seen some other species that we didn't know occur in some areas. And how are we combining this with the genetics? We are using a genetic technique that's called environmental DNA. And as its name says, it's basically taking or grabbing an environmental sample and detecting every little piece of DNA that's on it. So imagine you go and grab 1 liter of water in the beach or in a lagoon or in a river, and then you take that to the lab, and in the lab you isolate all the tiny little cells or pieces of tissue or any potential source of DNA. What kind of DNA is in a liter of water? Well, every organism releases DNA. When we lose a little bit of hair, or when a tiny bit of skin falls off. In the case of the whales, they release a lot of skin. In the case of fish, they pee, they poo. So every little organism is somehow leaving some DNA in the environment where they live. So what happens if I take this sample of water back to the lab and I detect, let's say, whale shark DNA? What does that mean? That means that in the place where I collected the water, there was a whale shark in a recent period of time. And why this is important? Because we are not capable, we're not able to be underwater snorkeling or diving day after day, month after month, year after year, right? We can just go from time to time and do this. So by taking the environmental sample, I can find a lot of animals that are difficult to spot, that are not always easy to see or easy to count. So this helps us, again, to identify all the diversity in a specific place. But this is something we're just starting with. Overall, our main interest, as I mentioned at the beginning of this talk, is to support the conservation. And I'm very, very happy to take any questions if you have some. I just wanted to end with a slide of the team that works here with us. It's this is something you cannot do alone, as many other types of research. This is something for which you need a whole team. We have a lot of students, we have a lot of volunteers. We even have a program with kids where we take them to the field so they can learn what we do and how we do it. So it's a lot of hands doing a little bit. So I with this, I don't know if you have any questions.
Wonderful. Thank you so much, Diana. That was awesome, and really great to see the work that's happening, that you're doing out in the Galapagos. It's a place that I've always dreamed of going, and you're not making it easier for me, because seeing all the photos and the amazing work is just making me want to go even more. So if anyone has any questions, please put them in the Q&A box. I'm going to start off by asking something that I ask everyone that comes to do the webinars for us. What is your favorite species of sharks?
That is the hardest questions of all. But if I have to pick one, I would say is the hammerhead shark. I wasn't sure before, but the time we encountered the first baby hammerhead, which is like this big, like just like a stuffed animal, I completely decided that was my favorite shark.
Oh. Yeah, hammerheads are one that comes back quite often. They are really, really special animals. Um, in fact, you talk about there being a lot of your projects are on baby sharks, which is really amazing. The Galapagos must be such an important place for these populations of sharks and rays. And how do those baby sharks have any predators? And how do they avoid them?
Yes, they do. So one way they avoid predators is they use these mangrove areas that are not easy to access for big sharks. So that's what makes our work a little bit easy, that we find them in shallow waters and we find them in areas that we can access, but where other big sharks cannot. And they stay for these for quite some time. They spend up to 6, 8, 9 months in these areas until they're a bit bigger. When they're a bit bigger, they adventure to go outside because they can probably deal better with predators. And the other way they do, we have been finding out, is the areas they use. They sort of camouflage. We found this with the rays, right? So the eagle rays, which are dark colored, black, they do prefer stay in areas that are rocky. So they are not very easy to spot. While the golden rays, for example, that have this gold color, they do stay on sandy areas. So they're not very easy to spot either. It's difficult to find golden rays over rocks or eagle rays over sand because they'll be an easy prey. So that's another way we're finding out they avoid predators.
That's awesome. Yeah, those golden rays are really beautiful. You were saying that they once they leave the mangroves, once they get big enough, they leave the mangroves and they leave those protected areas. Do we know where they go, and do they ever come back?
We know for some species, for the hammerheads, for example, we know because in the case of the hammerheads, we're not only studying the babies, but we're studying the juveniles and the adults, and we've been following them up. So we know when they leave the mangrove areas, they go to these isles, to these rocky isles. So these tiny little pieces of land that are not too far from the coast, big island. And they spend another couple of months there. And then when they're a bit bigger and they're in a big group, then they adventure to go farther to the northern islands, for example, which are over 100 kilometers away. And when they get a bit bigger, then they move even to other countries. For example, they go to Cocos Island in Costa Rica. And then eventually, yes, they do come back. They do come back when they are grown up, and the cycle just repeats.
Awesome. So it's the same. So the sharks that were born there come back and give birth to their babies there.
Some do, not all of them. And that's one question we're still trying to respond. Why is it that just some of them have the babies here and the others go to Costa Rica to have the babies and just come to feed and to hang out? But that's something we're trying to figure out.
Really? That's really interesting. Awesome. We have a question in the comments that is going into a bit more about how humans are affecting these sharks, and a lot of a question about illegal shark finning in the Galapagos. Is there a much happening? Is there much illegal finning or fishing of sharks? And how has COVID affected all of this?
Those are all great questions. First of all, so shark finning, it's prohibited by law here in the Galapagos for the last 25 years. So that it's not happening legally. However, the marine reserve is small, and outside of the marine reserve, we do see shark finning happening. Unfortunately, that's no longer a protected area, so and it's not under the jurisdiction of the country. So that's something that's very hard to manage, and something that happens. I don't think only in Ecuador, but all over the world. Um, However, I was telling you also that these nursery areas are very important for sharks and for rays. And although there's no shark fishing and shark finning, we do have other activities, for example, tourism, which of course is not as shark finning, but um, if when we have a lot of pressure by tourism, we can destroy the habitats because we have a lot of boats coming to the mangrove areas, because we have a lot of people swimming and stepping on the rocks, on corals sometimes. So we are somehow destroying the habitat. And for this, there are strong regulations here in the Galapagos for the number of tourists that are allowed to go in one area or another on a single day. So you cannot just go anywhere. But that's something we need to keep an eye on, because it's something that sometimes it's not seen as a risk, but it is. And how has COVID impacted? Well, it has for sure stopped tourism for a year. So that's something, it allowed the ecosystem, it allowed the islands to have some sort of a break and to recover a little bit from all this. But the activities have started to come back, tourism starting to come back. I hope we have learned from this, and we have realized that we need to give this break to nature sometimes, that we need to stop on all the pressure we're putting.
Oh, yeah, that's right. People often only think about um, the fishing and finning when it comes to the impacts that we're having on sharks, but a lot of our activities will have different levels of impact, indirect impacts through affecting their habitats or the water that they're living in. So I guess when it comes to tourism, we might have some people watching us who want to come to the Galapagos one day to see the amazing wildlife and everything that the Galapagos has to offer. They might want to come and visit one day. So what would you say someone who wants to come and visit can do to minimize their impact on the environment and on sharks?
Well, yes, you can be a responsible tourist, that's for sure. You can make sure you don't just put on garbage on nature. You can make sure you're reusing your water bottle. You can make sure you are not bringing plastic bags. You can for sure be part of this, and you can transmit that message and share that message with other people that are surrounding you, right? We often think the problem in the sea starts in the sea. Well, not. The problem with the sea starts at our homes. It starts with the decisions we make on how we are protecting this, right? So you can also learn, you can read and learn about what Galapagos has to offer. So if you come to the Galapagos, then go and do the activities that are following all the regulations of the national park. If the regulations say do not go out of the trail, do not feed the animals, do not touch this, then follow the rules, because the rules are there for a reason, right? They are there to protect the animals and also to protect ourselves. And the Galapagos has plenty to offer. If some of the people who's here will ever want to visit, please come and please enjoy this beautiful place.
Wonderful. Yeah, that's right. Whenever and that really is something that people should do wherever they go, whenever they visit somewhere, whether you're a scuba diver or someone who just likes to go on hikes or who likes to visit villages and historical sites, always inform yourself before you go and visit somewhere to make sure that you're following the rules and respecting the environment, the wildlife, the people. So that's really good advice. We have a question following up from that in the Q&A that wonder what would be the impacts on the Galapagos if tourism stopped? Would it be positive or would it be negative? Because maybe that would be a surprising result.
We just, actually, we do have a response for that because COVID stopped tourism for a year. So that's something that we never imagined that would happen here because it's such a touristic place. Yeah. But then a pandemic came and everything had to stop. Zero flights, zero tourism, all businesses shut down. And yes, that has an impact on the local population, and a terrible impact because most of the people here live, they depend on tourism. So like 80-something% of the people work in something related to tourism. So that means a lot of families lost their income. Lots of family were suffering a lot because nobody had a job. There was no money coming in, and that comes with problems, of course. What are you going to eat? What are you going to do if you do not have a job? So the solution is never something as radical as we think. Zero tourism, nobody is allowed to come here because we have a population living here that depends on these resources. The solution is more about the responsible management of such resources. We want to take advantage of this. We want to enjoy, we want to have tourism. We can do, but we can have this properly regulated. So a specific amount of tourists visiting a place. So not having 500 people stepping up the same trail in 2 hours, but having a fraction of that. So we know the animals are not getting overstressed because of seeing so many people. We are reducing the probability of someone just dropping garbage on the trail. We're reducing, and it's easy to manage also groups that are smaller, to control groups that are smaller, and to educate these groups as well.
Wonderful. Yeah, thank you so much for sharing that. I guess we've not got any more questions coming through, but I just wanted to ask you one last one before I let you go, which is a bit more of a personal one and more about your work and your field work. What has been a moment on field work or in your work in general that you've really had an amazing moment in your field work or in your work? What has been a moment like that for you where you've really thought, wow, this is awesome?
This is awesome. I would have to say it was the same, finding for the first time a baby hammerhead shark, because that was completely unexpected. And that was also like, you see, I've seen these animals before. I've seen the adults, I've seen the big ones, and they look so powerful, so strong, so everything. But then watching a tiny baby and realizing how vulnerable they are and how easy it is for them to get harmed, that was, yeah, something that completely changed the way I was seeing these areas and these animals.
Oh, that is so special. Baby sharks just are just so cute, and they just, yeah, I remember seeing baby nurse sharks and baby lemon sharks for the first time, and it's really hard to resist the urge to just kiss them on the nose there. Well, Diana, thank you so much for joining us and for sharing your work with us and for answering our questions. We really, really appreciate it. Do you have anywhere that people can follow your work or the work of your organization, and maybe keep up to date with all of your results?
Yes. Well, I have a Twitter account in the university. So I work for a university and a research center here. I'm going to put it on the chat. I don't know if everybody sees the chat.
Yeah, they should do.
Okay.
And
That's the name.
if anyone has any last minute questions, make sure that you just pop them in the Q&A. Otherwise, you'll lose your chance. I might just have to copy and paste it to attendees.
Well, that's the name of the center. If you look it like that in either Twitter or Instagram or Facebook, you will find it, the Galapagos Science Center. And that's like a channel where we are posting not only the research of sharks and rays, but the research with sea lions, the research with plants, the research with turtles, with tortoises, with everything. And then I put my Twitter account there as well. I do share more about sharks and rays specifically, but I highly recommend following the center because it's a bit of everything.
Awesome. Yeah. Thank you so much. I also definitely recommend following them. They put some great content out there. Again, thank you so much for joining us, Diana. And if anyone wants to find out more about sharks and rays and it wants to rewatch the webinar or any of our past webinars, you can check out our website, sharksforkids.com, and you can follow us on our social media. This talk will be on YouTube maybe in the next couple of days, so make sure to share it around as well. And next time we have, I believe it's Frida joining us, Frida Lara, which is really exciting. So make sure that you join us for that one too. And with that, I will say goodbye and thank you again, Diana.
Thank you, Jenny. And yes, go and attend Frida's seminar. It's awesome, and she will have tons of amazing pictures to share.
Thank you, Diana.